All right, well, we are just at the top of the hour, so we'll go ahead and start today's webinar. My name is Sarah Burris. I'm the scientific liaison here at Fujifilm Visual Sonics. We have a really exciting presentation for you today titled Shortwave Infrared Photoacoustic Imaging for the Study of Alzheimer's Disease. Our speaker, Chris Salinas, is a graduate research assistant at the University of Arizona, working in the College of Optical Sciences. Just a few notes about our webinar. A recording of the webinar will be made available once we finish today. As a reminder, all the lines are muted for the duration of the webinar. So if you have a question, please use the Q&A button located at the bottom panel of your screen. Questions will be answered at the end of the session, and you can expect our webinar to last approximately 30 minutes with another 5 to 10 minutes of questions. So without any further delay, our speaker today, Chris, has earned his Bachelor's of Science in Physics from the University of Nevada in 2020. That same year, he joined his current research laboratory with Dr. Russ Whitty at the University of Arizona, where he acts as the lead researcher in creating novel shortwave infrared photoacoustic imaging techniques. During his time with Dr. Whitty, he's refined the capabilities of their imaging systems by integrating various signal corrections and denoising algorithms in data processing. He's currently co-leading the creation of various optoacoustic coupling media to use in clinical machinery. He also acts as a team lead for a multi-university biomedical imaging study observing the healing effects of a novel laser-activated skin sealant membrane. In June of this year, he joined the U.S. Army at the RTI Imaging Technology Branch as an optical engineering intern working on a classified project. He has already gained authorship on several publications and hopes to finish his PhD in the next 2 years. And with that, I'll turn it over to Chris.
Awesome. Thank you so much for that introduction. So today we're going to talk about shortwave infrared photoacoustic imaging for the study of Alzheimer's disease. This is something that my group has started working on within the last 2 years, and we're pretty excited about the overall direction of where this project is going and its applications towards potentially helping the study of Alzheimer's disease. So to get into this, for the quick presentation outline, we're going to start with background and motivation. Particularly, we're going to focus on lipid and water contrast in this presentation. So I'm going to talk about the role of lipids in Alzheimer's disease and what are some current challenges and current problems with imaging lipid in the brain. And then we're going to go on to the goals and the proposed solution and how Visual Sonics and the Vivo Laser X can actually be used to solve this challenge of lipid imaging, lipid and water imaging in the brain. We'll go into sort of the experimental methods, the spectral unmixing theory, how to calibrate and test our model, and then we'll talk about some preliminary results, some results from various samples and also some preliminary results imaging a mouse brain, looking at what we believe to be distributions of gray and white matter in the brain. So getting into this more, let's talk about a little bit of background on Alzheimer's disease, lipids and water. So Alzheimer's disease, I'm sure everybody has some familiarity, whether that's through personal anecdote or just in general. Alzheimer's disease is the leading cause of dementia in the world, and it's estimated to affect around 20 to 50 million people worldwide. It is known that deficiencies in the transport of lipid and plaques in the brain and their metabolism increase the risk factor of Alzheimer's disease. And so this sort of indicates that structural changes of lipids in the brain may play a vital role in, let's say, detecting early onset Alzheimer's disease. And it's also known that atrophy, or let's say loss of brain volume, is a known factor as well. So these are all things. This is certainly not the whole picture of Alzheimer's disease, but just for today's discussion, let's just focus on lipid transport and lipids, and let's say water volume or brain volume of the entire brain. So moving on to that, let's talk about the current ways that lipid is imaged in the brain. So this mostly consists of MRI and optical methods, which all can have various limitations. Let's say MRI. You can have lower resolution, up to 1 millimeters, long acquisition time, very expensive equipment. With optical methods, we have very high resolution, but only at very superficial tissue depths, a few millimeters in general. You can see on the left, we have some MRI images. Each column is separated by 1 year, and you can see some atrophy in the hippocampus area of this human brain from non-Alzheimer's to experiencing symptoms of Alzheimer's. And you can see, obviously, there's some atrophy and some movement here in the hippocampus area. And the resolution, maybe it's good enough for this purpose, but certainly if you wanted to know anything more, it might not be sufficient. And the specificity here, it might not be ideal as well. On the right, you can see some vasculature of the hippocampus area. This is through OCT. And while the resolution here is very good and it's specific, you can only get, I believe this study is only going down to 1.5 millimeters in depth. So there's this sort of unmet need for a non-invasive way of measuring lipids at good specificity and good resolution, let's say around 100 microns in resolution at a depth of 5 millimeters. So there's this gap between MRI, good field of view, low resolution, and OCT, good resolution, low field of view. There's this sort of gap which motivates the application of photoacoustic imaging. And I'm sure we're all somewhat familiar with that here, but we see this as a great gap to bridge for this topic and actually motivating the use of photoacoustic imaging. So with that, now that we've set sort of the motivation for why we want to use photoacoustic imaging, let's talk about some challenges with lipid and water imaging and what's sort of already been done in that regard in that subfield. So the main problem with imaging, specifically lipid, is this typically used photoacoustic range of 680 to 980 or 600 to 1,000 nanometers. You can see on the right with the absorption coefficients, lipid has very, very low absorption, something that you might not even be able to detect with a lot of systems. Now, if we expand our waveband search, let's say we go into the shortwave infrared, these are going to be your idler frequencies on, let's say, the Visual Sonics Vivo Laser X, which I'll talk about in a second. But if we expand our search into the shortwave infrared, we can see that lipid, the absorption increases a lot, and it actually goes into the range of what we can actually detect with many photoacoustic systems. Now, keep in mind, and you can see on the graph here, water dominates absorption past, let's say, around 1270 nanometers. So what this means is, as we go past, let's say, around 1270 nanometers, water is going to be the dominant absorber of photoacoustic signal in tissue, and that is going to inherently limit our depth penetration. You can't see it on this graph here, but if we go past 1400 nanometers up to 2000, the water absorption is incredibly strong, and this is going to limit our depth penetration. So we have to be careful if we want to image lipid and water with photoacoustic imaging. We do have to be careful with the specific waveband and the specific wavelengths we choose, because we don't want to go too low where there's no contrast, but we also don't want to go too high because maybe there's a strong lipid peak at 1770 nanometers, but the absorption of water is also simultaneously so great at that wavelength that we're not going to get much depth penetration. So our overall objective, knowing the motivation, is to use photoacoustic imaging to map lipid and water content with a greater contrast and depth penetration within certain spectral windows of the shortwave infrared, or the SWIR. Now, going on to our approach, how we're going to solve this is we're actually going to make use of the Visual Sonics Vivo Laser X. This is a great system. I've worked on it for, I think, a year and a half now. I don't really have any complaints about the system. There's a lot of things you can do with this. You have access to idler frequencies, so that's a tunable laser between 1200 and 2000 nanometers. Real-time B mode and photoacoustic imaging capabilities. We have a range of high-frequency ultrasound probes. The ones we use in my lab are 20 to 50. I think we also have a 40 or 44 megahertz probe as well. This system allows you to export the data, the raw data, into something like MATLAB or Python, where we can actually manipulate the data ourselves and fully take control of the unmixing, which I'll explain a little bit later on. And it's very easy to use, of course. So our overall approach for mapping lipid water content in tissue would be to modify the setup a little bit for transmission mode imaging, which is light from above, ultrasound from below, and I'll talk to that, I believe, in the next slide. Then we want to construct an optimal imaging sample compartment. Then we're going to take full spectrum data, multi-wavelength data, combine that all, and then really see if a spectral unmixing method can bring out contrast between lipid and water. And of course, we're going to be operating in this shortwave infrared, 1200 to 2000 nanometers, for reasons I said last slide. So now going on to what I mean by that transmission mode setup. This means light coming from the top of the sample, illuminating the top surface of the sample. You can see here on the left, we have the laser bundle. And an important note here is that on the laser X, we're using the green laser bundle. And that was chosen, I believe the width of this laser bundle is about 1 centimeter. And this was chosen just to fit the dimensions of our compartment and the dimensions of the samples we were predicting we were going to image. You can see that the sample fits right here, and let's say it was 1 centimeter by 1 centimeter compartment. For this figure, this is just a brain, but this can be anything from a muscular tissue sample, a small chunk, 1 centimeter by 1 centimeter, at least in cross-sectional width and length, a phantom, a brain, etc. But moving on, we have light illuminating the top surface of the sample, and it's important to note that this path length of this laser is just through air. And that is done to maximize light penetration in photoacoustic signal. Now, this is a problem I'll be talking about more in the conclusions, and this is also some exciting work we're doing related to this talk, but it's important to have the optical path through air because traditional acoustic coupling medium, let's say agarose or ultrasound gel, are mostly water-based. There are some rubber-based ones, but the ones I'm speaking of are mostly water-based. And if we want to image in this 1200 to 2000 nanometer range, we cannot have our optical path length through water because we're going to get this sort of adverse absorption from the coupling medium before the optical fluence even hits the sample. So we have to be sort of stringent on this. And again, this is stuff we're working on, and I'll talk about it a little bit later. But it is important to note that the path length is through air. And while the optical path length is through air, we have some contact with the sample and a thin, I believe this is saran wrap or some plastic wrap with a little bit of water coupling here. And there's a water reservoir in contact with an ultrasound transducer on the bottom. So this allows for acoustic coupling below the sample, optical illumination of the top sample. And for this study, while we do have access to the 50 megahertz probe, for this, we're going to use the 25 megahertz linear rate transducer from the Laser X. This was chosen because of the field of view, the depth of field of this probe, and sort of the fact that later when we want to do in vivo studies, 25 megahertz might give advantages above the 50 megahertz for penetrating the skull of a mouse if we're looking deep into the future about actually imaging Alzheimer's. Yes, so continuing on. Now we'll talk about now that I've sort of talked about the setup, let's talk about the wavelengths that we're actually going to use. I was saying before, we have to be very meticulous about deciding where we're going to do our unmixing because we don't want to go too high and limit our depth penetration, even though we might get better SNR, we'll only get penetration on the levels of OCT, which is something, our motivation is moving away from. We also don't want to go too low because there's no contrast. So we've sort of been playing around with this and we found some good results, which I'll show in a second, but let's look at the wavelengths of 1230, 1270, and 1360 nanometers. You'll notice that at around 1230 nanometers, the absorption coefficients of lipid and water are identical. So what this allows us to do, and this is sort of a clever trick, we can use this to help our depth dependence fluence correction. We know that photoacoustic signal is proportional to some constants like the Greenesian parameter, temperature dependent constants of the tissue. Then we have the two main factors in this signal is the absorption coefficient and the laser fluence. So if the laser fluence, if the amount of light penetrating the tissue is less and less, this means that we need to do depth correction to correct for the lower laser fluence. And if we do depth correction to 1230, where the absorption coefficients align, this allows us to exploit based operating on the assumption that most of what we're imaging is either going to be lipid or water-based. We can make the assumption that most of the tissue is going to have around this same absorption coefficient, and we can do our depth correction that way, operating on that assumption. We can talk about if that's a good assumption to make, but we believe with the types of samples we've been imaging and we've been testing for and calibrating, it is a good assumption to make. So key takeaways here, 1230 nanometers, the absorption coefficients are the same, and we're using that to use our depth to do our depth dependent fluids correction. Now, going on to the actual spectral unmixing, we have data at 1230, and like I said, we're also going to take data at 1270 and 1360. You can see on the bottom right here with these absorption coefficients, if we start with a baseline image at 1270 and we compare that differentially to a baseline image at 1230, we would expect no increase in water signal to the contribution of the overall differential image. But however, lipid, we have a 9 or almost a 10 times increase in signal. So if we take a differential image at 1270 and 1230, we expect most of the differences to be from a lipid. And the same can be said on the other side. If we have a baseline image at 1270 and we compare that with one at 1360, while lipid stays constant and even below probably the ability of our system to capture, the water increase is dramatically. So on either side of this 1270 nanometer baseline, we can get an estimate of how much lipid there is and how much water there is. And we can do some cross correlation, some stuff on the back end. But what this leads to is what we call the lipid water saturation. And this is, we've made this so this is completely analogous to a blood oxygen saturation in literature. So one thing that the Laser X and the Vivo Laser X does a great job at is its oxy-deoxy-hemo unmixing, which is something that we wanted to take from and move into this lipid water unmixing as well. So now we've made a lipid water saturation, just like blood oxygen saturation. Excuse me. So now that we've set up all of that baseline, let's talk about some results. So first order of business is we wanted to see if our unmixing method actually does the job that it's supposed to. So we created a phantom. This is made with a mixture of half and half and lipid shortening. What we did was we took two phantoms, one with mostly water, and then one with a 25% increase by weight in lipid content, and we stacked them on top of each other. And we put them in our compartment to verify that the separation layers can be seen with our unmixing method, and that we can actually predict quantitatively a interlayer difference in lipid percent that's accurate to our a priori knowledge of what it should be. And so you can see on the left here, we have a diagram. We have a water layer and a lipid layer. Again, this is made from half and half and lipid shortening. I believe agarose is the material used to sort of solidify and make these sort of slabs of gelatin-like material. In the middle here, you can see our lipid and water unmixing map. So the darker blue color in this represents a higher water concentration, and the darker red color represents a higher lipid concentration. And this green is something that we've been working on recently. This is exploiting the fact that we're using 3 wavelengths to do our unmixing. So technically, we can have a third component here, maybe a noise component that we can throw things in and bin things in, and that is represented by the green color. But for this, you can see that, especially in accordance to the diagram on the left, we have a clear separation of a water layer and a lipid layer. And this is something that on the B mode, it's not trivial to see. Maybe there's a little bit of contrast in the B mode, but when you use this method, you can clearly see we have regions of high water and high lipid content. And this looks very similar to, if any of you have done oxy-deoxy-hemo unmixing with the Vivo system, this looks very similar. The color scheme is almost the same. And from this data, we can get statistics such as mean concentration variance and all that. And we can actually see the distributions of each region. So on the left here, we see that this is the distribution of water from here, which is expected. It's mostly water. There are some emulsifiers and things like that in there, but this is mostly water content. And on the lipid side, we can see that the interlayer difference here is about 29%, which is in the range of what we expect it to be. We made it around 25%. So this is in the range, and this was sort of an interesting technique to sort of see if our unmixing method was working and see if there's any adjustments we need to make. So going through that, we end up approaching the imaging of locally sourced bovine meat. This was actually what was awarded the Vivo Research Award, I believe, in 2021, which sets the basis for this. So the next slide you can see here, this is the same sample with different cross sections. On the left, you can see, left and the bottoms, you can see a diagram of locally sourced bovine meat. These are cut into sections of 1 centimeter by 1 centimeter cubes. And these chunks of meat were chosen because they have lines of intermuscular fat. So you know what marbled meat is, or a good steak, you have these lines of intermuscular fat separated by chunks of lean meat, which is mostly going to be water-based. So here we can do a 3D scan. And here we have a depth width cross-section, or lateral and axial cross-section. And you can see on the ultrasound, while there's not much contrast here, we can see that with our unmixing method, we can really bring out areas of high lipid and low lipid. So this is, again, looking at regions of intramuscular fat in this chunk of lean meat. And on the right, we have another cross-section, longitudinal and lateral. And once we get to lipid, we can really start to see, again, this is just the same sample, we can really start to see that we can predict with some quantitative accuracy the percent lipid there. So this is some stuff we've been really excited about, some stuff we've got some great results from. Now let's bring it back to our motivation for the Alzheimer's disease. We do have some preliminary results. We have obtained a mouse brain. Now this mouse brain, let me preface this by saying this mouse brain in particular that we've only imaged one mouse brain so far, and this mouse brain was healthy. It did not show signs of any Alzheimer's. It was actually used for a traumatic brain injury study, I think about 2 or 3 years ago. So this might not be the ideal brain. It's not necessarily a fresh brain, but we make do with what we have. So building on all of our results, we've now just recently started applying this technique to the actual imaging of a mouse brain. You can see the underside picture of a mouse brain here, where we show different regions of gray and white matter. And at this moment, we believe most of the contrast generated in our images, when we apply this shortwave photoacoustic lipid mapping, is bringing contrast from regions of gray and white matter in the brain. You can see a relative PA spectrum. We've taken raw PA spectrum data of both what we believe to be gray and white matter sections in the actual brain. And on the right here, you can see a lipid water overlay map with the corresponding B mode image on the bottom. And this is pretty interesting. While on the ultrasound, maybe you can see there's some sort of contrast here between dark and light regions. If we apply our lipid water map, we can really start to see, oh, okay, that's probably more lipid-based tissue, maybe something closer to white matter. And we also have this good correlation between the boundary of the air and brain boundary here, where it's mostly going to be water-based tissue on the surface of here. And some more data here. This is the same brain. These are not the same slice of the brain, but the left column is the brain imaged right side up, where the dome of the skull is facing the upper side of the image. And then the right column is where we invert the brain, and we want to see if we can test the depth accuracy of our unmixing method here. So you can see again on the bottom row is the raw B mode images of the corresponding images above, and the top row is the lipid water map as well. And while we can quantitatively analyze these regions of predicted high and low lipid content, and we can compare that to what we would expect gray and white matter to be. And particularly with white matter, we obtain data that is pretty much spot on with what the accepted value is for lipid concentration of that white matter. The gray matter estimations are a little bit off. We are not quite sure if this is due to the fact that this brain has been sitting in saline for months on end, and this brain is not necessarily fresh, but regardless, we're intrigued by our estimates of the white matter lipid content. We can see that this is sort of invariant to the orientation that our sample's at. We are with our depth correction algorithm, we get more or less the same value, 58% if we invert the sample versus 57% if we don't invert the sample, which might be some evidence to suggest that our depth correction is doing a good job. And this is, let me say again, this is something that's very new. This has just been recently done, so there's still a lot of work that needs to be done. With this, we still need to do a proper noise analysis on our photoacoustic images and spectrum to really confirm that what we're seeing is potentially gray and white matter. And if we move on to our conclusions, building off of that, just to wrap sort of things up, a 3 wavelength unmixing technique was used to identify lipid and water content in tissue. So we first did testing on a phantom where we knew where we know the predictions of lipid and water. And we use the unmixing method to distinguish and discern the interlayer differences. And what we found was the interlayer difference between lipid and water percentages were in the range of what we expected them to be. Then we moved on to sections of locally sourced bovine meats where we can really see, not so well in the ultrasound image, but when we add the lipid map, we can really start to see these streaks and lines of intermuscular fat that exist throughout the tissue. And then building off of that even more, we've just recently started applying this to the mapping of a mouse brain to potentially show what we believe to be regions of gray and white matter at this time. And some preliminary data we showed some agreement with the predicted lipid concentrations of those two regions, but certainly more work needs to be done, and that's something we're very excited about. So this all shows that our unmixing method, we believe, has promise in discerning lipid content at practical depths of 5 millimeters. We got to think back to our motivation. OCT, while having better resolution than us and probably being a little bit more specific, can only get you a few millimeters in depth. And this 5 millimeters mark was chosen because you can image most, if not all, of the mouse brain at that depth, or you can do an inversion and certainly get almost all of the mouse brain there. So this whole thing is something we're very excited about, and this lends its potential to the future study of Alzheimer's disease, different stages of Alzheimer's disease in mice models. And some future work, we do want to investigate the lipid structure in mice models with varying degrees of Alzheimer's disease. We do want to do a lot of further validation for our method, anatomical and composition. We want to take images with an MRI machine and compare what the gray matter and white matter distributions look like there, and compare that to what our algorithm is predicting. Particularly with the atrophy of the brain, when we think about Alzheimer's, we can, like I said in the introduction, we think of atrophy. And if what we're seeing is more or less similar, that would be a great comparison, again, cross comparison with MRI and/or histology. And then another end goal that we've been simultaneously working on is the engineering of this towards a reflection mode imaging system. So all of these studies, as I stated, were done in transmission mode, where we have the light illuminating the top surface and the ultrasound probe beneath the actual sample. Now, while that's good, it's not necessarily ideal. We are losing a lot of our photoacoustic signal as the acoustic signals travel through the actual sample and then through the water coupling medium. So the acoustic path length is not ideal. While the optical path length might be, the acoustic path length is not. So the motivation for reflection mode imaging would be to increase our SNR and to also make it more, easy to use for the user. If they can just do the standard reflection mode imaging with the probe that most everybody does, I believe, with Vivo Laser X, they'd be able to do this very easily and very quickly, and they don't have to manipulate the setup in any way like we have done. We do have some materials that we've been studying, that we've been developing, that will allow this reflection mode to happen, and this consists of having materials that allow acoustic waves to travel through, but also shortwave infrared light to also travel through. We do have a provisional patent on that, but I'm not necessarily at liberty at this time to disclose any more of that information. But yeah, I think with that, that's all I have for today. So thank you everybody for listening, and I'll be happy to answer any sort of questions you guys have. Thank you.
Great. Thank you so much, Chris. It was a really, really fascinating talk.
Okay.
A few questions have started to roll in here through the chat. Can this method be adjusted for use in a reflection mode system?
Yeah, so that's a good question. With the reflection mode system, while we would predict to have a better SNR, we would need to adjust the unmixing method slightly because, and I'm trying not to disclose too much information, but we would need to adjust the unmixing method slightly because there is some slight absorption of the optoacoustic coupling medium that we need to take into effect. The absorption of the coupling medium is much, much less than what it would be from, let's say, a standard old, but it is something that we do need to take into account. this to a reflection mode imaging system.
Great, thank you.
Mhm.
A question for better photoacoustic imaging, what should be paid attention to in the design of organic molecules?
What should be paid attention to in the design of organic molecules?
Mhm.
I'm not sure I understand the question. What should be paid attention to?
So I'm
Mhm.
I could be wrong in this, but I'm thinking maybe they're referring to in the fact that if you're not imaging in water and you're kind of looking at this, subsequent medium that you're going to try to image through, are there special design considerations or things that you've already ruled out that you absolutely could not use in that capacity?
For imaging through,
Like in the absence of imaging through water, since that's not really an option when imaging lipids.
Yeah, I think with our design for this, there's definitely many things that we need to take into account for if we do want to put in an acoustic coupling medium for the reflection mode imaging. There are many things that we need to use, and there's been so many trials and error. I mean, so many nights in lab where things aren't working, of course, but yeah, there are many things, particularly density, speed of sound, certainly optical absorption, Fresnel reflection. All those things need to be paid attention to when we're trying to develop a medium that we're trying to image through to make this thing more easy to use and make it a reflection mode imaging.
Great, thank you. If the person that requested that question would like to email Chris afterwards, I'm sure he'd be happy to continue the conversation with you. If we didn't actually successfully answer that here. In some of the images that you showed close to the last, 2/3 of the presentation, the audience has commented that there was no continuity of water and lipid content in the images and the larger ROIs after unmixing.
Yeah, this
were
one,
the
right?
images thresholded?
Yes, so these images were thresholded. I don't have the threshold off the top of my head, but yeah, this is an interesting point. And I'm glad people brought this up. This sort of tells us that we need to do more validation on this method to make sure that what we're actually seeing corresponds to regions of gray and white matter. Now, one could look at this and think this might be due to an artifact of noise, either produced on the photoacoustic end or on the unmixing side of things. But yeah, you're right. There might be some, but overall, I would agree there's not really any continuity, which sort of tells us that maybe this is more of an artifact noise and we need to do some more cleaning up of this. But that is something that we are aware of and sort of motivates us to do much more validation before we can bring this to a point where we feel confident that we can actually use this to study Alzheimer's disease. Again, this imaging with the brain has been very recent. And yeah, this has only recently come about in our lab. And yeah, we definitely need to do more validation with this. Mm-hmm.
Great, thank you. Can you please provide some clarification on the composition of the dual layer phantom study? One user was specifically asking about designation of half and half water, since half and half with water kind of seems to be two very different substances.
Yeah. So we're using half and half as an emulsifier to actually help bind the lipid and water together so that we can easily mix them and create more or less a homogeneous area or a homogeneous sample. We didn't really want to have regions of really high lipid where the water and the lipid didn't mix, and we just wanted as a consistent sample as possible. So while we did add water and half, we did have to use half and half as an emulsifier to bind the water and lipid. And this is something that my lab has done before, looking at thermoacoustic imaging, where we've made little tubes of differing lipid and water, and we've chosen half and half just to be an emulsifier. And yeah, the spectrum of that, we do have, I didn't present that today, but we do have raw spectral data for the half and half, and it's not exactly water, which is sort of corresponding to why this distribution is only, let's say, 75% water. But half and half, to answer the question, was used as an emulsifier to help bind the lipid and water together to make a consistent sample.
Great, thank you. Can you talk a little more about what kind of unmixing techniques you've been using?
What kind of unmixing technique has been used? Well, so for our case, we're doing a 3-wavelength unmixing technique, looking at the differential images between 1230, 1270, and 1360. There have been other studies that are looking at things like this, and most everybody is particularly, let's talk about collagen as well. Collagen is also an absorber in this range. I guess to answer this question, to highlight why we're using this 3-wavelength unmixing is really just this spectral window where we believe we are going to have the best penetration. And also, while we might be sacrificing some SNR, not looking at let's say the main absorption peak of lipid at 1270, we do have much more depth penetration, which will allow us to hit this 5 millimeter mark that we've set, so that we can potentially image a whole mouse brain.
Great, thank you. And it looks like just maybe one more question here. Are there other applications where discerning lipid versus water might be beneficial?
Yeah, let's say atherosclerosis, so looking at plaque in arteries. I think that would be a great way. The unmixing method might have to be a little bit different because you'd be going through a lot of blood, so oxy and deoxyhemo, but that is something where a 5 millimeter penetration depth could be good, either maybe in vivo or maybe invasively, non-invasively. But there are certainly many diseases where lipid contrast and having the ability to discern lipid contrast would be beneficial. So atherosclerosis might be one.
Great. We actually had 2 more questions come in through the chat during that. Would it be practical to image lipids at 940 nanometers where there is a small peak?
Yeah, so that's a great question, and it is somewhat practical, but let me go back to this slide here. Yeah, so there is a small peak right here at 980, but the main issue with that would be SNR. And I do see the next question would be about the skull limitation. So when we think about doing this in vivo, actually, our SNRs might be really sacrificed. So while it can be done, we really want to maximize the signal that we're getting from lipid, especially if our images, as we've sort of discussed before, on the brain images might not necessarily be very accurate at this time. There might be some noise artifacts and everything. So we really want to try to maximize the signal. So while it can be done at 980, we're really trying to really maximize our signal while also imaging in areas where water might not be that absorbing. And we have done some studies where we're looking at 980 and then going up to 1770, where there's another really big peak of water. And we've sort of found that our best bet here is going to be in this 12 to 1300 nanometer area. And then I guess moving on to the next question I see with the skull limitation, yeah, absolutely, that would, for some reasons I just suggested, that would be one. But this is also why partially why we've chosen to use the 25 megahertz probe for this study. While the 50 megahertz probe would get us obviously better resolution, if we're thinking about in vivo traveling through the skull, the use of the 50 megahertz probe might degrade the quality of our images and the absolute acoustic signal that we get through the skull. So using the lower frequency, the 25 megahertz probe, was used with the thought of this could potentially be in vivo in the future, and we need to take data on a probe that would be realistic for taking in vivo data, which for the visual sonics, I've been on many studies where we do take in vivo data using the 25 megahertz probe and the photoacoustic capabilities of the Vivo Laser X.
Wonderful. Well, thank you so much, Chris, for your time today. Again, really, really amazing presentation. Exciting to see what you're doing with the Laser X, kind of taking this one step further than kind of common publications that we see coming out with photoacoustic imaging. For the audience, thank you as well for joining us today. Just as a reminder, if you have any questions for Chris, please send them over to us and we're happy to forward them over to him. And please remember to connect with us on our website. We have a variety of resources available to you, not only recordings of past webinars, but resources in terms of imaging guides, video tutorials, and a variety of information that will help you in all of your imaging studies. So thank you again. We'll see you next time.
Thank you, guys.